Hi everyone, I'm Francine Marques, Professor of Genetics at Monash University in Australia, and I serve as Deputy Director Discovery at the Victorian Heart Institute. Last month, in December 25, I was invited to deliver the Colin Johnston Lecture at the Hypertension Australia annual scientific meeting. And after some feedback, I was asked to record this lecture and make it available online for others to be able to watch. So there it is. I hope you enjoy it. So in 2025 marked 20 years since the Colin Johnston Lecture was established, and I am incredibly honored to be this year's lecturer. This honor is deeply personal because Colin was my PhD supervisor, which makes me in some ways his scientific granddaughter and my team his great-grandkids. That lineage is something that I carry with pride. There are three lessons that my supervisor, Brian Morris, taught me that he learned from Colin, which I wanted to share with you today. The first is to be personable. The second is to be well connected and help others make connections. And the third is to develop well-rounded researchers. And these lessons have shaped not only my career, but also the way that I mentor and I lead my team and in their careers too. For those less familiar with history, I just wanted to briefly remind you that the High Blood Pressure Research Council was formed in 1979 with Austin Doyle as the chairman, Paul Korner as the secretary, and Colin Johnston as the treasurer. Even though I trained decades later, I still had the privilege of benefiting from the lessons through the wisdom that was passed on by others. I first came to Australia in 2006 after finishing my Bachelor of Science, my honors, and my master's in genetics and molecular biology in Brazil. And I came to have a gap year, and that was 20 years this year. I then was offered a PhD scholarship at the University of Sydney with Professor Brian Morris, that you can see here in the middle. And as I mentioned, who was a trainee from Colin Johnston. After that, I was offered the opportunity to do my first postdoctoral training at Federation University with Professor Fadi Chacha in collaboration also with Professor Stephen Harrop and also Maciej Tomaszewski from Manchester University, who I also had the opportunity to spend some time with in his lab. And during this time, I learned many lessons from these three people, but I wanted to share specifically something that I learned from Stephen Harrop, who was one of Austin Doyle's mentees. And Stephen always said that Doyle taught him to do research in both animals and people. And I hope that you see through my presentation today that we have honored that tradition. I also then had the opportunity to move to the Baker Heart and Diabetes Institute, where I had two of my key mentors that helped me there. So Gavin Lambert and also Jeff Head. And Paul Korner is a former director of the Institute, and he was a very important mentor to Jeff Head in particular. And through this mentorship, I also learned to think about hypertension not in isolation, but as part of an integrative physiology. That perspective, that seeing hypertension as a multi-system disease, is ultimately what led me to explore the gut and its role in cardiovascular health. And during this time at the Baker, that's when I had the opportunity to train under David Kay and also collaborate with Professor Charles McKay, starting the work in the gut microbiome field. And finally, in 2018, I was recruited by Professor Mauro O'Brien to become a lab head at Monash University. And more recently, in early 2025, we then relocated our lab to the Victorian Heart Institute. And one of the key things that I learned during this period, and I'm going to share some lessons during my talk that I think it's important for junior members in the community, is that it is important to have mentors. And also, once you have had mentors and you have advanced your career, make sure you're paying it forward. So the vision for our lab is to improve cardiovascular health by building exceptional scientists. And we do this by harnessing the microbiome as a new non-traditional risk factor for high blood pressure and cardiovascular disease. And one of the key things that I also have learned in the past 7 years as a lab head is that my team, the culture in the team, and the people in particular, they are number one in everything that we do. And for people that know me, you know how passionate I am about talking about research culture. And that is my lesson number two. And this is particularly for senior people that are listening, is that the most powerful investment you can do to advance your research is to invest in your people first. And with that, I want to acknowledge my team members that were present with me at the conference. There were 9 of them, 7 that got selected as finalists for awards. And I want to specifically acknowledge Charlotte Barker and also Rikesh Mullaritharan, who won the awards for the student and the early career researcher oral presentations, which are some of the most prestigious awards at the Hypertension Australia meeting. And before I tell you where we are now, I want to take you back in time when I started this research back in 2015. And at the time, I had some personal challenges in my life that drove me to change my research direction. And that led me to think 3 things. The first is that I wanted to focus on primary prevention, and hypertension is a great tool for that because it is the most prevalent and also important underlying factor for common diseases. Also, the microbiome offers a fantastic translational opportunities, and I wanted my research to move more into a translational field. But also, I wanted to give more back to community, and our community can relate to and loves talking about food. What I learned in the past 10 years doing this research as well is that people not only love talking about food, they also love talking about their biohabits, and I'll tell you more about that later. But I came across some challenges, and the first one is that nobody had fecal samples from patients, and that meant that I had to go on and recruit my own participants to start my own sample collection to build that. The second is that nobody knew how to analyze the microbiome, so I had to go and learn how to do that myself. And the third is that we didn't really talk about the microbiome in cardiovascular health and research. And that meant that we basically had to establish a whole new field. So my third lesson is be open to evolving your research identity and also have a growth mindset. That's not because you or someone else hasn't done that before, that doesn't mean that you're not capable of doing it. And with that, we started to work on the microbiome. So we have our microbes or the microbiota. So these are the microorganisms that live in an environment. And then we also have the microbiome, which has the microbes, as well as many other environmental factors, such as, for example, the metabolites that are in that environment, as well as environmental conditions. And we now know that we have a similar number of bacterial to human cells in the body. And if we would then count other types of microorganisms, we actually have a higher number than human cells. And the vast majority of these live in our large intestine or our gut. And we have many knowns and many knowns unknowns in the field. A field that I want to highlight is, for example, that at this point, when we sequence the microbiome, we can only map about 80-85% of those sequences, so we still don't know what all those sequences do. And we have over 300 million genes in that microbiome relative to the 20,000 human genes. That means that our microbiome has many more genes and does many more things potentially. We also have then one of the key other things is that diet is a key factor that drives the microbiome. Well, we also have many known unknowns, and that means that there are many hard to detect microbes such as viruses that we're still only beginning to understand. And we also have a lot of genes that we still don't understand the function. But more importantly, and perhaps important to this audience, is that what microbes actually cause common disease and how they do it is still something that we're trying to understand. And when we study the microbiome, there are many layers, and it's not just about specifically the microbes. So we can study microbes directly by culturing them, but this can be proven difficult, particularly with the ones in the gut, because many of them are anaerobic, meaning that they would die under exposure of oxygen. So that makes very tricky to culture them. But there's some methods, but that's harder. So what we tend to do more is just sequence their DNA or the metagenome, because that tells us who is there and what genes they have. We can also study them at the RNA level, looking into what genes they express. or at the protein level, understanding what proteins then are being produced. And this is particularly interesting because this allows us to look into the human, the food, and the microbiome proteins, not just the microbes themselves. After that, we can also look into the metabolites, and this is important because these metabolites are the really key that facilitate that microbiome to host communication. And this is important because these metabolites can then cross our gut epithelium barrier. They can leave the gut itself and then come into our systemic circulation, where they can activate host receptors and downstream signaling mechanisms that ultimately lead to changes in phenotypes such as regulation of blood pressure. So what does the microbiome then have to do with hypertension? Well, hypertension is what we call in genetics a multifactorial disease. This means that we have genetic and epigenetic factors as well as environmental risk factors that in isolation they are well known. But what we still don't understand really is how these two are interacting. And back in 2018, in a series of papers, we proposed that as a part of this was being regulated by our gut microbes. And we're really excited to see back in 2021 that the Page's mosaic theory of hypertension, a key theory that combines all the key aspects that regulates blood pressure, was redesigned to include the microbiome as one of the key factors that is involved in blood pressure regulation. Now we know that it is indeed linked to every single other aspect of blood pressure regulation, also culminating in an American Heart Association Science Advisory that was published in July 2025, which purpose is to give specific insights into prominent, evolving, and scientific issues that are of interest to the public and health professionals. And our contribution has been twofold, looking into humans across cohort studies, clinical trials, and population-based studies to understand what the human hypertensive microbiome looks like, what these microbes do, whether we could predict cardiovascular disease based on these microbes, and how we can harness them to then reduce blood pressure and cardiovascular disease. But then also using animal models as these are key for proof of principle interventions, as well as understanding what factors that are regulating the microbiome and pinpointing specific mechanisms. And through that, to this date, we have now recruited 5 cohort studies and clinical trials. We are custodians of data from another 3 clinical trials. We have worked with 7 knockout strains, and we have published over 90 papers, including 6 guidelines, some of them shown here, that have been crucial in advancing the field. And one of the key things that I learned in this process is the importance of engaging with our community, because we need to think as a stakeholder, identifying problems that need solving, being that from a clinician side or from a patient side as well. And we have been able to do this taking 2 approaches. The first is understanding the human gut microbiome in hypertension. And a key differential of all of our studies is that we recruit and diagnose all of our participants using 24-hour blood pressure monitoring, which is gold standard in the field. And through that, we have, for example, determined that people with hypertension have different microbial pathways in the gut. We also determined that these microbes are associated with blood pressure variability, and that we can predict long-term cardiovascular disease risk based on proteins that are being produced in the gut by either our tissue or our microbes as well. And also that we look into people that have high heart failure with preserved ejection fraction, which is a disease that results from many years of uncontrolled high blood pressure. They have a very distinct microbiome relative to healthy people from the community. We also have looked into or moved further this recent years into a more integrative analysis of microbiome, the immune system, and blood pressure. So, for example, looking into how we can integrate this multi-omics analysis, pinpointing mechanisms that are independent of animal models, as well as showing that we have many different microbes in the gut that are associated with activation of different patterns of immune cells in our systemic circulation. So, for example, here CD4 and CD8 positive T cells that we know that regulate blood pressure. Our second approach has been about understanding the role of dietary fiber on blood pressure. And this is important because the first evidence that dietary fiber lowers blood pressure came from a study that was published in 1979. And we have more recently had many meta-analysis and clinical trials showing that indeed high fiber reduces cardiovascular incidence and mortality. And a key factor for that is a reduction in blood pressure that you can see in these two studies. But a key question that remained in the field was exactly how this was happening. And this is something that we have been able to pinpoint in a series of studies over the past 10 years. So my lesson number 5 is seize opportunities. Look back in history or think about the future, because there are many problems, for example, that were first identified in the 70s, in the 80s, that we didn't have the technology to answer back then, but we do it now. Or think about what are the key challenges that we're facing going forward in the next 3 decades that we need people to start thinking about solutions now. So we have been able to help understand how dietary fiber reduces blood pressure. And this was a crucial paper that was my first paper in the field that is now a highly cited paper that has helped in this process. And what we have been able to pinpoint is that dietary fiber does this via our gut microbiome. So the way that this works is that when we ingest fiber in our diet, we don't have enzymes that digest fiber in our in small intestine, meaning that fiber always reaches our large intestine intact. And there is, as I mentioned before, where we have the highest amount of commensal bacteria in the body, and certain types of fiber can be fermented by this bacteria. This fermentation process releases as byproduct metabolites called short-chain fatty acids, primarily acetate, propionate, and butyrate. And these have a localized effect in the gut, helping us to improve our gut barrier. And this is really key in maintaining our microbes there and reducing inflammation in the body. But also they can be distributed to the rest of the body, where we then show that they could lower blood pressure in animals. But we're also interested in understanding whether we could do this in people. And to be able to answer this question, we performed a randomized clinical trial that was crossover, placebo-controlled, double-blind, and so absolute gold standard. And we saw that a 3-week intervention with a high short-chain fatty acid supplement called HEMS-AB was able to significantly reduce 24-hour systolic blood pressure by 6 millimeters mercury, which is equivalent to taking 1 medication. We have now done further studies and developed a new type of fiber using a grant from the Australian Stroke Heart Research Accelerator. And we're hoping that this is a product that we may be able to commercialize in the next 5 years. But we also have done some further research in this, showing that people that were likely to respond to this diet, they had an overall reduction in 24-hour systolic blood pressure of 10 millimeters mercury. And they also started the trial with less fiber intake. So that's a quick, easy way to try to reduce your blood pressure, is to increase your fiber intake. And we are currently recruiting for the Grains BP trial. If you're interested, you can have a look on our website, more information, and we should be still recruiting for that during at least the first semester of 2026. And this leads me to my lesson number 6, that is that make sure you challenge yourself and also surround yourself with great collaborators from a diverse background, because the type of research that we are doing and the questions that we still need answered in cardiovascular health or in medical research overall, they're not questions that can be answered by just one person or one set of expertise. So having diverse and collaborators and expertise is really crucial in advancing any type of medical research. So we then went on to also study how the short-chain fatty acids lower blood pressure. And what we have been focusing is on G protein coupled receptors as key receptors that facilitate the interaction between short-chain fatty acids and then blood pressure lowering effects. And we have focused on 2 sets of receptors. The first are short-chain fatty acid sensing receptors. So these are receptors that directly bound to short-chain fatty acids and activated by short-chain fatty acids. And the second set of receptors are those that sense pH changes because these are acidic metabolites. And I'll briefly tell you more about this. Very briefly, probably will summarize about 5 or 6 years of research into a couple of slides. So what we determined was first that when we have a lack of GPR41 and 43, even on the high fiber diet, this leads to higher blood pressure in a mouse model. What we determined then next was how this happened. And we identified that these animals, they go on to have a breakdown of the gut epithelial barrier that you can see here in the cover of the journal. And this leads to basically passage of bacterial products such as lipopolysaccharides or LPS into the gut barrier. This leads to LPS binding to TLR4, which is a receptor on the surface of our immune cells. And this can then activate inflammation. So what we did was to give these animals a TLR4 antagonist, so blocking this process. And as you can see, the blood pressure changes were then restored. And what we did next was to indeed show that this is immune dependent by doing reciprocal bone marrow chimeras. So what we did was to take the immune cells from the wild type animals and give to the knockout animals and vice versa. And we can easily see here that that phenotype was swapped, particularly showing that activation of macrophages is really important in how then dietary fiber and short-chain fatty acids regulate blood pressure via GPR41 and 43. We also have done some analysis in humans looking into genetics as a way to show the involvement of these receptors. So what we did was to look into common genetic variants, showing that the variants that have that increase expression of both GPR41 and GPR43, they go on to significantly reduce the rates of hypertension in a large data set of 276,000 people from the UK Biobank, as well as looking into rare pathogenic variants. And these are important because these basically copy a human knockout. And this was done in a data set of nearly 400,000 people in the UK Biobank, showing that people that have this very rare but pathogenic variants, they go on to have higher odds ratio of developing hypertension and any form of cardiovascular disease, even if they have adequate fiber intake. We also then had a new hypothesis that in the end of the day, short-chain fatty acids are acidic metabolites, and as acidic metabolites, they would decrease intestinal pH and activate pH sensing receptors that then would lower blood pressure. And using the smart pill motility testing system that you can see in this figure. This is a system that monitors gastrointestinal motility and pH in real time. We identified that people that have hypertension, they have a significantly higher minimal colonic pH, meaning that they have likely less short-chain fatty acids being produced in the upper regions of the large intestine. And this manuscript is now actually available as a preprint. We also have been able to then understand which receptors are involved in this process, and we do it to the pH levels that were identified in the human gut. We have been able to pinpoint 2 receptors of this pH sensing family. GPR68 was presented in detail by Evany Dinakis at the 2024 hypertension meeting, where she won the Student Oral Award, and it is now published. So I'll focus more on GPR65 instead. So what we were able to show is that a model that lacks GPR65 is able to significantly increase blood pressure, especially, and this is abolished under low fiber conditions. So this is specific for when we have a pH that this receptor is activated on the high fiber condition. And this is again driven by immune cell activation, this time via CD8 positive T cells, where we then perform adoptive transfers to Reg1 knockout mice that don't have any B or T cells, showing that again we can increase the animal's blood pressure in a period of 2 weeks with these CD8 positive T cells. But we also have been interested in understanding for the prevention of cardiovascular disease. And prevention doesn't start just in our generation, it starts in the next generation. So we have been looking into the impact of high fiber and short chain fatty acids in the offspring and understanding how this happens. And what we have been able to do by looking into maternal dietary fiber interventions during pregnancy is show that this leads to cardioprotection in the offspring, showing that the offspring of maternal high fiber mothers had smaller development of cardiac hypertrophy. They were protected against development of cardiac hypertrophy, cardiac fibrosis, and this seems to be driven by epigenetic mechanisms such as changes in natriuretic peptide acetylation that seems to be prevalent still in the adult offspring. More recently, we also have looked into sex-specific changes, specifically looking into single-cell RNA sequencing, showing that these changes that affect the offspring, both female and male offspring, that are related to immune cells, but this anti-inflammatory effect seems to be more prevalent in male offspring. And if people are interested in this field, I suggest reading a major review that we have published recently in this topic. But the last part of this talk, what I want to tell you about is then what happens when we don't eat enough fiber. And this is because globally we're having about 11 grams of fiber a day, while recommendations are more than twice that. So a minimum 25 grams a day for women and a minimum of 30 grams a day for men, and even higher levels if we're talking about hypertension. And the estimation is that 4 out of 5 people in Australia have inadequate fiber intake, but I also have seen others propose that globally it may be maybe 19 out of 20 people instead. So low fiber diet doesn't only contribute to cardiovascular disease, as I will show you soon, but also contributes to constipation. And what we found is that constipation also increases the risk of cardiovascular disease. So when we looked into the UK Biobank in 400,000 people, we see that even adjustment by traditional risk factors and medication, we see that people that have cardiovascular disease, people that have constipation, they have higher rates of cardiovascular disease. And in particular, when we look into people that have hypertension, if they have also constipation, they had a 34% increased cardiovascular disease risk. And as I mentioned earlier, people love talking about the bowel habits because this was our most mentioned article ever published in the community. So we reached nearly 100 outlets, had a very high Outmetric score. There was a lot of community engagement because people love talking about it. But in a more serious note, we're also failing to recommend fiber intaking guidelines. I came across this by accident that I was trying to find how much we're recommending in guidelines, and then I realized that none of the guidelines actually did recommend it. So I put this paper, this table together in particular to highlight the fact that we have a lot of evidence showing that dietary fiber lowers blood pressure, and it still fail to mention it in guidelines. And as Stephen Harrop told me recently, in an auction, all you need is one buyer. So I'm hoping to show this here and hope that my one buyer is in the room and can help us drive this to guidelines. And my lesson number 7 is this, that no matter who you are or what your expertise is, make sure you advocate for issues that matter to you, because it is possible to change the field if you really believe in it, really put effort into driving this. And with that, I do want to mention that I'm really excited that these guidelines for fiber intake was then taken into the new American Heart Association guidelines recently. So I hope that the Australian guidelines and all the international guidelines will follow. And we started interest in this work about low fiber intake and the impact on cardiovascular disease back in 2020 when we published this paper showing that indeed low fiber intake increases blood pressure. And this happens via our gut microbes, that even if we transfer these microbes to animals that don't have any microbes, the blood pressure increases spontaneously. But what we didn't do at that study was to actually identify how this specifically happened. And our aim in the past 5 years has been to be able to identify the microbial metabolites that increase blood pressure when we don't consume enough fiber. And I'm very excited to tell you that I think we finally know what is happening. So what we first did was a metabolomics analysis to identify potential metabolites. And with that, we identified P-crystal-derived metabolites, particularly P-crystalglucuronide or PCG and P-crystal sulfate or PCS that are high when we have low fiber intake in animals. And this also increased the phenylalanine, tyrosine, and tryptophan biosynthesis mechanism. And when we give these animals antibiotics, what we can see is that the levels of these metabolites are abolished. We also wanted to understand the impact of translation for this. And to be able to do this, we looked into two cohorts. The first in-house cohort showing that people that had inadequate fiber intake had higher levels of PCG and also higher levels of the phenylalanine, tyrosine, and tryptophan biosynthesis mechanism. But also in the Twins UK cohort with our collaborations in the UK, showing that indeed low dietary fiber intake is associated with higher PCG levels, and PCG levels also associated with hypertension. But as we all know, association does not equal causation. And with that, we have spent a lot of time then trying to show that indeed high PCG levels caused hypertension. We started doing these experiments using animal models, and for many different reasons that I'm happy to discuss another time, these experiments failed. And my lesson number 8 is sometimes things are going to go wrong, but we eventually tend to get them right. So what we did was then to turn into genetics again and use a new technique called Mendelian randomization that basically relies on the random segregation of alleles to, similarly to a clinical trial, dictate associations. And what we found was indeed that PCG increased systolic and diastolic blood pressure, showing that there is indeed a causal association between PCG and increased blood pressure. So this takes me to my lesson number 9 is be resourceful. If one way fails, find another way. And we also have then leveraged these human data sets to look into potential mechanisms by doing co-expression analysis between RNA sequencing in the blood and PCG levels in the Twins UK cohort, showing that PCG is negatively associated with many anti-inflammatory pathways in humans. And finally, we wanted to understand whether we could reduce PCG and how fast that would happen by looking into the samples from our clinical trial that fed people a high fiber diet enriched with short chain fatty acids for 3 weeks. And what we can see is that some of the key metabolites that changed were indeed P-crystal and PCG, showing that it is possible to reduce the levels of these metabolites in as little as 3 weeks. So by now, you're probably thinking how you actually reduce your cardiovascular disease risk by leveraging dietary fiber and the gut microbiome, and what is the best approach to do it. And I'll tell you, it is not via doing fancy fecal microbiota transplantations or taking antibiotics. It is a food is medicine approach. And the evidence suggests that the best reduction of blood pressure and cardiovascular disease comes when we have a minimum of 10 servings of fruit and vegetables a day, and also 3 servings of whole grains a day. Overall, this is going to lead to an increase in fiber intake. And we know that every extra 5 grams of fiber a day reduces systolic blood pressure by 2.8 millimeters mercury. So we need to be aiming for a minimum 25 grams a day for females and 30 grams a day for males. And if you're thinking also how you improve your gut microbiome, well, it's also about diversity. Studies point that if we need to be having a minimum of 30 different types of plant-based foods per week to have a higher number of diverse microbes, that also higher number of short-chain fatty acid producers. So how do we do this at the population level then? Well, there's a study that looked into the global burden disease study. I looked into the diet-related deaths specifically and found that these accounted for 16% of the total deaths and 37% of cardiovascular deaths. And when we looked into the key dietary risks, what we see is that the number one is not salt, as many people might have predicted. It is actually low whole grain intake, followed by low legume intake, and then the third one is a high sodium intake. So how do we actually increase whole grain? Well, I want to propose to this audience the Danish Whole Grain Partnership, a program that was established in Denmark in 2008 that involved 28 organizations across government, foundations, food industry, manufacturers, retailers, researchers, community. And what they were able to do with simple measures over a period of 10 years was increase twice the amount of whole grain intake in the whole population. This means an extra 5 grams of fiber a day. And I'm not a health economist. We do have a new PhD student starting this year who is going to be looking into this. But for now, what I did was to ask Copilot to estimate what impact that would have in Australia. And it estimated that this would prevent about 50,000 cases of cardiovascular disease and save the economy $1.1 billion in direct healthcare savings in the next 5 years. So this seems a lot, and I really wish that's something that we could talk more about and potentially try to implement in Australia as well. So the key take-home message is that dietary fiber, if you want a happy microbiome, you need to be having high fiber because that's going to lead to production of short-chain fatty acids that act via G protein couple receptors, then lowering blood pressure and cardiovascular disease risk. While if we're having a low-fiber diet, that is going to lead to changes in our gut microbiome that increase peak cryptoglucuronide production, that increase inflammation and drive an increase in blood pressure and cardiovascular disease. So the key take-home message is that interventions to target the microbiome are promising new therapies for hypertension. And with that, I want to acknowledge everybody that has been involved in these studies, starting with our team, also our collaborators, our consumer advisory panel members and participants, and of course our funding bodies, and leave you with my lesson number 10, that work with people you like, because that's going to make your life much easier, happier, and much more fun. Thank you.